So in this lecture we are going to discuss the mechanism of muscle contraction and the sliding filament mechanism of muscle contraction. Mechanism of muscle contraction and sliding filament mechanism of muscle contraction. But before going into detail, let's recap and summarize what we have discussed in the the last lecture. We discussed that if we take a muscle and we cut it into two parts and look at one part, we will see that it will consist of small packets or pockets known as the fasciculi. One will be known as fasciculus and multiple will be fasciculi. These fasciculi will contain the muscle fiber, which are the muscle fibers are basically the cells, the basic cells of the muscles. So above the muscle there is a connective tissue cover known as the epimysium, and above the fasciculus there is also connective tissue cover known as the perimysium, and then above the inside the fasciculus there are muscle fibers. This is a fasciculus, and inside the fasciculus we see there are muscle fibers. So above the muscle fiber there is also a connective tissue layer known as the endomysium. So the endomysium goes and goes and connects with the connective tissue of the fasciculus, that is the perimysium, and then it goes and connects with the connective tissue of the whole muscle, that is the epimysium. So the epimysium, perimysium and endomysium, they combine to form the muscle tendon and attach the muscle with the bone. This provides attachment for the bone. So overall in the muscles we have packets of fasciculi. Inside the fasciculi we have the muscle fibers, and if you zoom in the muscle fiber with the help of electron microscope, we will see that a muscle fiber contains a lot of myofibrils. So this is one fiber which contains a lot of myofibrils. So this is basically showing one myofibril. This myofibril basically consists of a dark and a light zone, the dark bands and light bands. The dark band is known as the A band, while the light band is known as I band. I band basically consists of only light filaments, and or the A band consists of it consists of thin filaments. The I band consists of only thin filaments, while the A band consists of thick filaments and part of a thin filament. The thick filament, the A band was known as A band because it is anisotropic to light, while the I band is known as I band because it's isotropic to light. The thick band, the A band contains the thick filament and part of the thin filaments. I band contains only thin filaments. The I band has further been divided into two parts with the help of the Z line. The Z lines and the small part of I band from one side and a small part of I band from the other side, they combine with this A band, and the dead distance between these two lines, which are the two Z lines, is known as sarcomere. During the muscle contraction, the size of this A band, it is always the same, A for always. So the size of the A band is the same, while I band it decreases during contraction and it increases during relaxation. Then during a relaxed condition, there is a small space between the two thick filaments, and this is also a light zone, and which is known as the H zone. This H zone is present only when the muscle is in the relaxed state, and it is not present when the muscle is contracting. Then we discussed that the thick filament, they are basically made of myosin filaments. So thick filament are basically made of myosin filaments, while thin filaments are basically made of actin filaments. Then the myosin filament in itself is made of a lot of myosin molecules, and myosin molecules is then made of, each myosin molecules in this then made of 2 heavy chains and 4 light chains. So one thick chain is made of multiple myosin filaments, And the one myosin filament is made of a lot of myosin molecules. Each molecule, each myosin molecule is basically made of two heavy chains and four light chains. This heavy chain is making a tail of the myosin molecule as well as the head of the myosin molecule, while the light chains are only present on the head side of the myosin molecule. Then the actin filaments, or the thin filaments, are basically made of actin filaments. They consist of their basic structure is made of F actin molecule, and the F actin molecule is basically made of G actin proteins. There are two other types of proteins in the actin filament. They are troponin and tropomyosin, which we are going to discuss today. Each myosin is basically surrounded by 6 actin filaments, and the myosin molecule, the myosin filament, which is basically made of myosin molecules. This myosin filament, it connects with the actins with the help of cross bridges, and these cross bridges here, which can be seen here, it is connecting with the actin filaments. They are basically the heads of the myosin molecules, which are made of the heavy chain as well as 4 light chains. The Z lines here, which are basically forming the boundaries of the sarcomere, they are continuous from one myofibril towards the from one myofibril to other myofibril. For example, this is one myofibril and this is another myofibril. So this Z line is connecting the myofibrils with each other. This is, for example, one muscle fiber which is made of a lot of myofibrils. So all of these myofibrils are connected with each other with the help of these Z lines. So this Z line is not only determining the boundaries of a sarcomere, it also attaches these myofibrils with each other. Then, to maintain a distance between the actin and myosin, to keep them at a specific distance and keep them in a specific position, there is another protein known as titin. So titin is a very big molecule, one of the biggest protein molecules in the human body, and that is basically providing the basic framework, and that is providing the stability for the actin and myosin so that they can move in their respective compartments. Now, when we have discussed this whole thing, we will slightly discuss the sarcomere, and then we will move to sarcoplasm, sarcomere and sarcolemma, and then we will move towards the movement of the muscle fibers. So as every cell is having a cell membrane, similarly the muscle fiber is having a membrane. For example, this is one muscle fiber. This is the membrane of this muscle fiber, similar to the cell membrane. So this membrane is known as basically sarcolemma. This is the sarcolemma. This is basically the plasma membrane, and the endomysium is basically present on the outside of the sarcolemma. Then as the cell has a cytoplasm in which there is a nucleus, mitochondria, ribosome, etc. Similarly, inside the muscle fiber there is a cytoplasm that is known as sarcoplasm. So sarcoplasm is basically nothing but sarcoplasm is basically the cytoplasm of a muscle fiber. And then the sarcomere is basically the distance between two Z lines between which the movements of thick and thin filaments occurs. Then we will see that as the cytoplasm contains a lot of electrolytes inside the cell, and there are some electrolytes outside the cell. Similarly, inside the muscle fiber, outside the myofibrils, there are a lot of ions, and an organelle known as sarcoplasmic reticulum. As the normal cells has endoplasmic reticulum, the muscle fibers has a sarcoplasmic reticulum that contains a lot of calcium. So the sarcoplasm contains a sarcoplasm, which is the cytoplasm of the muscle cell. It contains a lot of iron like sodium, potassium, calcium, and ATPs, etc. And the sarcoplasm, which is basically like the endoplasm of the cell, it contains a lot of calcium. So what is the role of the sarcoplasmic reticulum, sarcoplasm, and sarcolemma in the functioning or in the contraction of the muscle fiber? We are going to discuss that now. Whenever a nerve signal comes to the muscle fiber, for example, a person is thinking like he wants to drink water, so he will have to move his upper limb as well as his lower limb. He will have to move towards a water cooler and take a glass and then drink it. So all these movements will need the movement of different types of muscles. So from where the signals are coming, basically the signals they come from the nervous system, because thoughts arise in the human brain, and the signals from the human brain, they are transferred with the help of neurons towards the muscles. When the signal reach the muscle, the neuron, for example, human brain, it's a human brain, and signal is coming from the brain through the spinal cord, for example, through a neuron into the muscle fiber. Normally each muscle fiber has its own nerve fiber that is coming into it, and the nerve fibers just ends above the muscle fiber. This is, for example, muscle fiber. In the nerve ending, this is the brain. The signal is coming from the brain, spinal cord, through a nerve, and the nerve enters the muscle, and they go deep into the muscle with the help of a special tubules known as T-tubules. The signal from the nerve from the above the muscle, they enter deep into the muscle fibers with the help of special tubules known as T-tubules. These tubules are basically helping in the spread of signal from above the muscle deep into the muscle too, so that they can reach the muscle fiber. Once the signal has reached the muscle fiber, the neuron, for example, this neuron is just ending above the muscle fiber. This is the muscle fiber. So this neuron will end here. You can see here a special neurotransmitter known as acetylcholine. This acetylcholine, it will get secreted. And this acetylcholine, it will act on ligand gated channels known as the sodium channels. These are sodium channels. Acetylcholine will act on these channels. These channels are present in the muscle fiber. The signal has come from the brain with the help of neuron, with the help of T-tubule. It has entered deep into the muscle fibers, and on the muscle fiber, on the membrane of the muscle fiber, it has secreted acetylcholine. This acetylcholine has acted on the sodium channel, and as soon as it acts on the sodium channel, a lot of sodium starts entering. A lot of sodium starts entering into the muscle fiber. This is myofibril. So inside the muscle fiber, there are a lot of myofibrils. See, this is fiber, and this fiber is basically having striation, light and dark zone, and these are present inside the muscle fiber. A muscle fiber contains a lot of myofibrils, and each muscle fiber, it is basically having one special neuron, or a small part of a neuron coming towards it, and which basically excite the whole membrane of this muscle fiber. So as soon as acetylcholine is secreted, it acts on the sodium channels, and sodium channels allow a lot of sodium to come inside, and as sodium is allowed and it comes inside, this membrane of the nerve fiber get excited, or the action potential is created on the membrane of muscle fiber. When the muscle fiber is activated, this signal goes inside, and when they goes inside, there is a special organelle known as the sarcoplasmic reticulum. Sarcoplasmic reticulum. This is whole muscle myofibril. Above here somewhere is a sarcoplasmic reticulum, which we have not shown in this diagram. Similarly, there is somewhere a neuron coming, which we have not shown in this diagram. So the signal has come and it has released from the sarcoplasmic reticulum a lot of calcium. Sodium came inside, it excited the membrane, and when it excited, this signal reached inside and it secreted a lot of calcium from the sarcoplasmic reticulum. As soon as this calcium is secreted from the sarcoplasmic reticulum, it acts on the thick and thin filaments and it helps in the contraction. As soon as calcium is secreted from the sarcoplasmic reticulum, it helps in the contraction, and this calcium then goes back. This calcium then goes back inside the sarcoplasmic reticulum, and it waits for the another signal to come, so that it comes out again and it helps in the contraction of the muscle again. When the calcium has been secreted, this calcium basically binds with the cross bridges or the head of the myosin, and it, suppose this is the head of the myosin molecule, it will pull the actin over here. It will start pulling the actin molecules, actin filaments or the thin filaments over it, so that the actins basically touch each other and they cross over each other. As we previously discussed, that normally there is a small space, there is a gap between the actins, but when the calcium has been secreted, it activates the head of the myosin. It starts pulling the actin towards each other. The cross bridges here and the cross bridges here. They all start pulling the actin, and the final result will be that the actin will move over each other. They will cross over. They will cross over each other. This mechanism of movement is known as sliding filament mechanism of movement, contraction. Sliding filament. Because the thick and the thin filaments, they are moving above each other, they are sliding on each other, so that's why this mechanism of movement is known as the sliding filament mechanism of muscle contraction. So that's all about the sliding filament mechanism of muscle contraction, and after this lecture we will discuss what are the qualities of the muscle, the myosin and the actin filaments, which and what are the basic parts of these proteins, and how their parts help in interaction with the calcium which has been secreted here, and how this calcium activates the heads of the myosin, and how this movement is basically occurring, how the heads of the myosin pull the actin over it, and how the sliding of the filaments occur. So let's recap what we have studied so far. The muscle, when cut and seen, it is made of pockets known as fasciculi. Fasciculi consist of fibers. The fibers consist of myofibril, and these fibers, due to the presence of myofibrils, they appear striated because of the dark and light bands. The dark bands are basically the A bands. The position of the A band is fixed, and the I band is basically a light band, and it is made of thin filaments. The A band is made of thick filament as well as small portion of the thin filaments, while the I band is basically made of thin filaments. When the muscle, when a signal comes through a neuron, the end of the neuron, it secretes acetylcholine. Acetylcholine activates sodium channel above the present above the on the membrane of the muscle fiber. This activation of the sodium channel allow movement of the sodium inside. Inside movement of the sodium causes activation of the sarcoplasmic reticulum. Sarcoplasmic reticulum releases a lot of calcium. Calcium binds with the heads of the myosins and the cross bridges, and it compels the pulling of the actin from both sides, so that these actins here start moving and they cross over, and the contraction of the muscle fiber occurs. Here you can see that this actin, this A band or the thick band or the dark band is just like the body of a bird or eagle, for example, while the I band is like their wings. So the body remains the same, the body remains the constant. The size of the body is not increasing or decreasing, but the wings of the bird or the wings of the eagle are increasing or decreasing in size, and it is helping in the flying of the bird. Same is the mechanism of the muscle. The A band size of the A band remains the same, and the I band increase and decrease due to the movement of the cross bridges due to the head of the myosins, and its size increase and decrease, and that helps in the movement of the muscles. So that's all about the sliding filament mechanism of muscle contraction, and in next lectures we will discuss the molecular details of the actin myosin filaments and their interaction with the calcium in ATPs. Thanks a lot for watching the video.